Okay, well, thank you, Lisanne. The next speaker is, as we went to the host, we're now going to go on the other side of the infectious arena, looking at the organism, Cryptococcus itself. And it's my privilege to introduce Dr. Andy Alspaugh, who's at our institution. Andy's a clinician, but he's a really true physician scientist, and he does work at the bedside and at the bench. I know he works at the bench because his lab bench is right beside mine. So he is going to talk on Cryptococcus, the fungus, and talk about it from that angle and how it produces disease. So Andy?
Thank you, John, for that introduction. It is a real pleasure for me to be able to talk with you again about the organism. All of us are interested in fungal diseases, fungal infections, and at one point in our lives, we were captivated by these microbes. And that's what I want to do for the next several minutes, is really look back at the organism to think about some of the microbial features that allow this particular organism to cause such a high burden of disease. And hopefully some of these are going to be informing the way we think about disease, about treatment, about novel therapies that we can envision for the future. I'm going to be focusing significantly on some work from younger investigators because I think they often have the best new ideas. But as we think about the fungi as a large group of biologically diverse and rich organisms, again, we know millions of fungi likely exist in the environment with several, maybe 100,000 with validly described genus and species. Why then such a small number of these in case reports causing human disease, and even a smaller number actually causing disease with any regularity, the ones that we're primarily focused on today? So I want to think about those features within Cryptococcus that allow it to cause such significant burden of disease, especially associated with HIV infection. But also we're seeing more and more cases in non-HIV associated infections. It's important to remember again that this organism, it grows typically as a haploid yeast. It does not need us in order to survive. So when we begin to think about these microbial features, many of these features really allow the organism to grow in the external environment. So the stresses that this organism is going to see, the temperature variations, its ability to survive solar radiation, its ability to survive nutrient deprivation and natural predators are features that are going to be allowing it to occur in its most common environment, just out in the world. But in contrast, as this organism is inhaled into the patient's lungs, many of these same features allow the organism to grow in us as well. And perhaps one of the reasons that this, among the many hundreds of thousands of fungi, is actually able to cause human disease. We heard a talk yesterday about the ability of an iron sequestering molecule potentially to be an effective antifungal therapy. And we see that in the host, it must be able to find those micronutrients that we try to sequester away from it. So it has efficient mechanisms for nutrient acquisition. Again, the same features that allow it to survive solar radiation likely allow it to survive nitrogen and reactive oxygen species assault that will occur inside the host. So what are some of the paradigms we can think about? As has been alluded to several times during the morning talks, fungi have this amazing, what I'm going to refer to as phenotypic plasticity. So they're not static organisms. They adapt with characteristic microbial trait changes in response to the environmental conditions. A couple of these are listed here. Normally growing in rich medium in the lab, even though we think of this as an encapsulated yeast, the capsule is actually expressed, at least on the cell surface, at a very low amount. And in contrast, when it enters host relevant conditions, we see dramatic increase in cell surface capsule. Similarly, the melanin pigment that protects it from reactive oxygen and nitrogen species in the host is an inducible change. And what is it about the organism that allows it this incredible phenotypic plasticity, the ability to change microbial characteristics according to the conditions? So I want to think about some of the molecular and genetic mechanisms behind phenotypic plasticity of Cryptococcus, some, as was alluded to with Dr. Perlin's talk, some of the features that allow it to really change its genome in remarkable ways, and how these two things sort of come together with what Lizanne was talking about with this new morphotype Titan cell formation. So many of these adaptive phenotypic changes are going to be the result of altered expression of various genes. And that can happen by traditional mechanisms where you have traditional transcriptional regulators that will bind and change the expression of genes. But I think one of the most exciting things that has come recently is the concept of the epigenetic regulation of gene expression, really by histone modification. So what does this mean? Many of you know, but DNA is really compact and tightly coiled onto DNA-binding proteins such as histones, and the relative openness and closed nature of the DNA will regulate whether genes are expressed or whether the DNA is able to be replicated. And this feature is really often regulated by the post-translational modifications of these DNA-binding histone proteins. So whether they're highly acetylated or deacetylated affects the openness and closedness of the chromatin. Now, why is this important? Well, again, the gene expressions will change, and we can change the gene expression by altering histone acetylation. This has been capitalized in anti-cancer chemotherapy. There are histone deacetylase inhibitors that will affect gene expression in certain cancer cells that are actually in clinical investigations right now. There are people looking at histone acetylation modifiers in HIV infection to drive the virus out of latency and thinking about cure. If we can get the latent virus out by altering histone modifications such as acetylation. And so Fabiana Brandão at the University of Brasilia was looking at chemical modifiers of histone acetylation and showed that pharmacological histone deacetylase inhibitors would regulate capsule expression in Cryptococcus. And then we went on and collaborated with her to do some genetic studies. There are 7 histone deacetylases predicted in the genome of Cryptococcus, listed here. And Fabiana went on to make mutants in all of these. And one would think with a family of similar enzymes that they may all be redundant, but they were not redundant. It seemed that these histone deacetylases had individual and definable functions that we could tease out by the phenotypes of the organism. So we see, for instance, in this mutant strain, we see capsule dramatically decreased. And in contrast, in this histone deacetylase mutant strain, we see dramatic increase in capsule. So there's this dynamic interplay of these proteins causing altered regulation of just this one phenotype. And you can imagine how this vast array of histone deacetylases can modify the DNA openness or closeness to help the regulation of many virulence-associated phenotypes. And she was able to demonstrate that single mutants were avirulent, completely avirulent. So you could imagine potentially targeting single ones of these enzymes might be able to be a potential future therapy. In addition to histone modification, we're talking about capsule expression here. We think about capsule being on the cell surface and how this may have an antiphagocytic role or somehow immediately modifying the local environment based on its binding to the cell surface. But some beautiful studies done by Jessica Brown at the University of Utah have looked at not only the bound capsule, but that this capsular polysaccharide is also secreted out into the local environment when it is not necessarily bound to the cell surface. And in this study, these dark images here, this is when she's probing for capsule polysaccharide. That's what these dark images mean here. And so you can see under certain conditions, we see a lot of capsular polysaccharide in the supernatant. And at other times, we see it bound to the cell surface. And there's an inverse correlation here, the blue being when the capsule polysaccharide was found in the supernatant, the yellow being when it was bound into the cell surface. So there are specific conditions when you see it on the cell surface and certain secreted into the environment. This is such a potent immunomodulatory agent that many of you know that this, when purified, can be used to induce remission of rheumatoid arthritis. So what would it be doing in the local environment if it's just secreted? This organism is actively modifying its local environment to affect the immune reaction to it. And so I think that and Jessica's done beautiful work dissecting the regulation of the secreted versus the bound capsule polysaccharide, and demonstrating here that when they actually see an increased immune infiltrate in the absence of the exopolysaccharide, and when you add exopolysaccharide, this secreted polysaccharide, you can decrease fungal burden and increase the immune infiltrate and the immune reaction to the organism. Now, what makes capsules stick to the cell surface? It's binding to the cell wall. And what we and others have demonstrated is that the cell wall is a tremendously active structure. This organism, depending on the environmental conditions, is continuously modifying and remodifying this in order to expose what it wants on the cell surface. What it doesn't want to expose are the highly immunoreactive molecules, the things that it tends to hide deep down in the base of the cell wall. And in contrast, it wants to expose the more immunologically inert things on the cell surface and potentially the capsule binding components on the cell surface. So there's a tremendous active remodeling. We have demonstrated that when we mutate, for instance, this transcription factor gene RIM101, that the organism is no longer able to actively remodify and organize its cell wall. This is the cell wall by transmission in, this very thick structure here that's completely disorganized. And we've also demonstrated that things that should be deep, such as chitin and chitosan, these highly immunoreactive moieties, you can see labeled here in green by wheat germ agglutinin, they're now present on the cell surface of this mutant where they're not present in the wild type. They're hidden from immune recognition in the wild type strain. And what does this do? Well, when the cell wall is disorganized and the chitin is exposed, we see that the capsule is no longer able to bind to the cell surface, and when placed into mice, we see this tremendous increase in the immune infiltration into the lungs of the infected mice, showing that the organization of the cell wall, capsule binding, immune shielding is very important in this infection. We looked at other mutant strains as well, and there was a very strong correlation between the exposure of chitin on the cell surface, as marked by wheat germ agglutinin, that correlated almost exactly with the degree of macrophage activation. So these immunoreactive epitopes on the cell surface completely drive this early innate immune response. Again, thinking about potential translational correlates of these cell wall changes that occur in response to infection. In Jenny Lodge's lab at Washington University, they have demonstrated that if they take and they start modifying some of these cell wall active genes, they create strains, again, with altered cell walls, and they can actually create strains that have induced protective immunity. Well, that's nothing for bacterial pathogenesis, but it's tremendous for fungi because we don't necessarily induce protective immunity by infection with Cryptococcus. And again, Stu Levitt's looking at different other cell surface components when he expresses them in the context of these cell wall sugars, he can also induce protective immunity. And just one of the graphs here, this is a lethal infection with Cryptococcus in an unvaccinated mouse, and you can see they typically die somewhere around 3 weeks after a pulmonary infection. And in contrast, when Jenny Lodge's group pre-vaccinated them with this chitosan-deficient cell wall mutant strain, these vaccinated mice, when re-challenged with the wild-type strain, complete protection in this model. So this is a dramatic result showing how important the cell wall is in preventing protective immunity, but maybe also with modifications able to induce some kind of protective immunity. So that's a little bit about the phenotype changes that this organism is exposed to in the context of infection. What about some genomic changes? So what are the changes in the genome? Now, I'm not talking about individual point mutations. I'm talking about large-scale genome changes. And some of these studies were really initiated by some of the work that David Perlin was talking about earlier today, So work that Jun Quan Chung had done about fluconazole heteroresistance. Now, any of you, who have dealt with Cryptococcus know that the concept of azole resistance was really not accepted for a long time, because once the organism was recovered in culture and serially passaged, it seemed that none of them seemed to have very high MICs against fluconazole. But when they began to study, if they took the organism out from a patient who had failed fluconazole therapy and they incubated in the presence of fluconazole, well, oftentimes they were able to find some resistant isolates. But when these resistant isolates were incubated under rich medium without fluconazole pressure, they lost their resistance with time. So something was happening. They were able to; these organisms were able to regain this fluconazole susceptibility. And what was this all about? Well, what June and her colleagues showed was that oftentimes there would be an acquisition of an entire chromosome, what's called aneuploidy, an alteration of the chromosomal content of these organisms, and it's listed here. So if the chromosome, this is chromosome 1, if it's a normal content, it will be somewhere around this line of unity, but if there's increased in any of the genes in chromosome 1, there will be this black bars, and you can see the entire chromosome is present in an increased copy in these fluconazole-resistant strains. It has added a chromosome. Now, we don't think of that as being a healthy state in humans, but apparently, as Dr. Perlman was saying, Candida and Cryptococcus, they can really alter their ploidy in amazing ways in response to environmental conditions. More recently, Łukasz Kozłowski at Clemson has redemonstrated some of these findings. And again, this is looking at flow cytometry DNA content, this being the normal haploid DNA content. And you can see the shift to the right in a diploid strain. Well, when you treat with fluconazole, you see the DNA content shift to the right. You see an increase in the ploidy of these strains. And what he demonstrated is that this mechanism happens because you have nuclear division without cellular division. You create basically a binucleate cell that eventually these sort of reorganize, and some of the chromosomes that aren't needed are lost. But in the presence of fluconazole pressure, chromosome 1 is almost always kept because it contains the target gene for fluconazole ERG11, and as well as other efflux pump regulators. And so we can see this amazing degree of ploidy plasticity, genomic plasticity, in response to various environmental stimuli. Now, both of these come together in this new, very important phenotype, the concept of titan cell formation. So for many years, people would see in the lungs of infected patients these larger cells, but they were assumed to be dead. They were just too big to be doing anything, in contrast to the normal cryptococcal size as seen lower in the panel. And again, one of the remarkable features, if you infect a mouse with Cryptococcus, this is an infected lung. Where's the inflammation? Little patches of inflammation here. This lung is filled with fungal cells. But somehow they're masking themselves from immune recognition. But many of these cells are quite large. This is a 50-micron scale bar. These are much bigger than the cells grown in culture. So there's this morphological transition that happens in the lung, where these very large cells exist. They are unable to be engulfed by phagocytes. They are resistant to reactive oxygen-nitrogen stress. They have large capsules. They're polyploid. They have 2N, 4N, 16N chromosome content, but then they can reduce back down to haploid again. So this organism is able to dramatically change its chromosomal content to create these states that allow it to resist immune clearance. This is important. In our treatment, these are more resistant to antifungals. Could these be persister cells? Could this be a site of persistence and dormancy, not just in the lymph nodes, but within the lung itself, where this organism is exposed to antifungals, is exposed to the immune system, but persists with time and serves as a reservoir for chronic infection? Several groups co-published ways to induce titan cell formation in the lab, in the test tube, which is important if we're going to understand its regulation. An in vitro cultivation system. This was done simultaneously by a group at the Pasteur, a group in Madrid, as well as a group in Birmingham, not UAB, but the other Birmingham, and in the UK. And this group with Liz Ballou, again, looking at that very large size of these titin cells, one of the fascinating features, guess what also induces titin cells? So stresses, but bacterial components. So I think that the interaction of Cryptococcus with other organisms it's going to find in the infectious environment is going to help, is going to signal that it is in an inhospitable environment. It will induce this phenotypic change to a more resistant persister state. So in conclusion, this is a remarkable pathogen. But one, again, that I think maybe has some chinks in its shield that we can maybe exploit. So it has tremendous phenotypic plasticity, but by understanding some of the mechanisms of the phenotypic change, perhaps we can direct new therapies. Again, its ability to alter its genome content so dramatically is a remarkable feature, and I think that this will be something that we're just beginning to understand. And then the recognition of these very different forms within the host is also going to inform our understanding of this disease. So with that, I want to thank you for your attention. I'm happy to answer any questions.
So, thanks.
Yes.
So presumably crypto didn't evolve aneuploidy so it could resist fluconazole. So what do you think the mechanism for aneuploidy is? Is this about xenobiotics that it would have encountered? And if so, what would have those xenobiotics been that are fluconazole-like?
Yeah, that's a great question. So so the idea that fluconazole isn't ubiquitous in nature, and this ability to change its ploidy has to have arisen from something else. So you're right. There are natural predators. There are other microbes that will secrete antifungal compounds. That could be part of the reason. I think that what we're probably going to recognize is that these ploidy changes will be a response, maybe a common response to many different stresses. Fluconazole being one. Fluconazole, what is the stress that fluconazole is adding? It's adding some kind of a cell membrane, ergosterol depletion stress. Are there other stresses in the environment that are going to similarly affect the membrane and its stability? Or so I think there are going to be any number of environmental stresses, that could be as simple as antifungal compounds secreted by competing microorganisms, or it could be something a bit more basic.
And so the other thing that we've seen with fluconazole is that you can get partial duplications, but not only, so chromosome 1's quite a nice and sort of tidy explanation because, as you say, the target, but you also see other chromosomes being duplicated. So do you have any idea about what the same sort of answer?
I think that probably what's happening is in this vast population, there are many chromosomes that would be left, and if it offers a competitive advantage, that population will increase within there. And so you think with Candida, they have this small isochromosome that's formed, but it's almost always this isochromosome 5 that has ERG11, has these sort of efflux pump genes on it. So it tends to be something in common that's directly related to the stress that it's seeing at the time.
On the same topic, I think we have it backwards. I think fungal genomes are really, really variable all the time. And the individual fungus that suffers a mutation or has gene duplication or chromosome duplication or whole nucleus duplication is lost because it's at a selective advantage. But if you put it under selection, like an antifungal drug, then you're going to find individuals, the ones that are successful, are going to retain that mutation that gives them an advantage. And early on in the debate about whether yeast had a whole genome duplication or not, this is early just shortly after Saccharomyces this genome was sequenced. There was a big debate, and one of the French were on the losing side of that debate. But in the course of that, they did an experiment where they could take a yeast strain that had one of the genes still duplicated and knock out one of them to make it haploinsufficient. And then they'd plate those out, and overnight they would get revertants. And when they sequenced those revertants, because they could sequence the genomes, it was big pieces of chromosomes, or whole chromosomes that had duplicated. So I think it's really common that fungi do this. They don't need any extra agent to do it. It's the selection that lets us see it.
I think you're right. And also, I think that we're limited by the models that we use. So, so much in terms of the cell cycle has been done in Saccharomyces cerevisiae, which has such a tight association between nuclear division and cell division. I mean, it's almost invariant. And in contrast, I think these other fungi, there's going to be a bit more looseness with that system, and maybe other models would be better.
Well, thank you, Andy. And I think it's just an elegant description of the yeast, Cryptococcus. And the word you take away from that is plasticity. The genome is very, very plastic.